Okay, so in this video I'm going to talk to you about the structure and function of starch, glycogen and cellulose. Let's start with starch. Starch is a polysaccharide. That word polysaccharide means many sugars. Starch is produced as a result of photosynthesis. So during photosynthesis, plants make glucose, and those glucose molecules are joined together via condensation reactions. They form glycosidic bonds between each other. And this is really important because glucose is soluble. And if we just left it as glucose within plant cells, it would just dissolve away and be lost and not be able to be used by the plant. But starch is an insoluble polysaccharide, meaning that it's a good storage molecule. So we find it in all plant cells, but plants store starch in their roots. So if you think about storage organs such as potatoes or turnips, these are basically big balls of starch. So it's really important for us because we eat potatoes, and so they form a major energy source in our diets. So starch is made up of long chains of alpha glucose molecules. Now, these chains can be branched or unbranched. The unbranched chains are made up only of 1,4 glycosidic bonds, and then the branched chains, they also have 1,6, so it's the 1,6 glycosidic bonds that make the branch. The great thing about starch is the unbranched chains, they form these tight coils, they form these helices that make it very, very compact. And this compact nature of the molecule makes it a really good storage molecule. So why is it really good at being a storage molecule? Well, it's insoluble, so it doesn't affect the water potential. Therefore, it doesn't lower the water potential of the plant cell, and so water is not drawn into the cell by osmosis. It doesn't diffuse out of cells. Because it's compact, lots of it can be stored into a really small space, and it can be hydrolyzed to form alpha glucose molecules. Now, one good thing about starch is because it's got these branches, it means that it's got lots of end points, and that means that there are lots of points on which enzymes can act to break off the alpha glucose molecules to allow it to be used in respiration, meaning that it's a very rapid release of glucose molecules, so a fast energy release from the starch. Starch is never found in animal cells, it's only found in plant cells. Starch is formed of two main subunits, amylose and amylopectin. Amylose makes up about 30% of the starch, whereas myelopectin makes up about 70% of the starch. So amylose is unbranched, and it's the bit that forms the helices, so it's the bit that you're most able to condense. So the great thing about amylose is it's a really condensed, compact storage structure, whereas amylopectin has branches, and so the good thing about amylopectin is it has lots of ends on which the enzymes can act, so it's a rapid release of glucose. So let's take a minute to have a look at the structure of amylose, which forms this coiled unbranched chain. So we can see that the coil is formed both by these series of 1,4 glycosidic bonds and also by these hydrogen bonds that are formed between the molecules in the coil that hold the helix in place. Let's look at amylopectin next. So you can see here that the branches are formed by these 1,6 glycosidic bonds between the 6th carbon here on this molecule and the 1st carbon here on this molecule. And this allows it to have branches, which increases the surface area for the action of enzymes. And that's pretty much everything you need to know about the structure of amylose and amylopectin. You'll remember from GCSE that the test for starch is iodine. So interestingly, iodine is not particularly soluble in water, so we have to add potassium iodide. You don't need to worry about that though. It's perfectly fine just to say the iodine test. So when we see the iodine at the beginning, it forms this sort of light orangey brown colour. And when we add it to a sample that contains starch, we will see a colour change, like a deep blue colour. This is because when the iodine comes into contact with the amylose coils, it fits inside the amylose coil and it causes that colour change. Interestingly, and you absolutely don't need to know this, when iodine interacts with the myelopectin, it actually turns like a purpley red colour, but you never see this because the blue colour that you see with the amylose totally overrides that purpley red colour. So what we see is that dark blue colour when iodine interacts with starch. So that's everything you need to know about the structure and function of starch. Let's talk about glycogen. So we know that glycogen is the main storage molecule in animal cells. It's really, really similar in structure to starch, so it's still made up of alpha glucose molecules, but it's more highly branched. So it's much more highly branched than starch. This means that it is a much more ready source of energy. So because there are more end chains, there's a much greater surface area on which enzymes can attach to remove the alpha glucose molecules for them to be used in respiration. This means it can be really, really rapidly broken down. So animals have a much higher metabolic rate than plants. They do a lot more stuff, so it makes more sense that their energy storage molecule would be more readily broken down. All of the other benefits of glycogen are really similar to the benefits that we earlier talked about with starch. So it's insoluble and it's compact. And the only one that's a little bit different is that it's more highly branched. Glycogen is found in a granular form in many cell types. Now, liver cells have the highest concentration of glycogen, with about 8% of the weight of the liver is just glycogen. That's about 100 to 120 grams. And then you've got glycogen in your muscles. There's way more glycogen in your muscles than there is in your liver. And you can also find a little bit in the kidneys, a little bit in your brain, a little bit in white blood cells. So you kind of dot it about where you might need it. And the average person has about 2,000 calories worth of glycogen stored in their body. So it's a really, really important energy store. And that's everything that you need to know about glycogen. Let's move on to cellulose. Cellulose is different from starch and glycogen in that, first of all, it's not involved in storage. It is a structural polysaccharide. And secondly, it's made up of monomers of beta glucose as opposed to alpha glucose. This seemingly small difference is actually super important because it totally changes the way that glycosidic bonds are formed, and therefore it changes the structure. So we talked about the fact that in starch and glycogen we form these coils, but because of the rotation, and we'll look at it in just a second, because of the rotation of the beta glucose molecules when they form glycosidic bonds, this causes cellulose chains to be long and straight. So they form these long, straight chains that run parallel to each other. And when they run parallel to each other, they then form cross-linkages via hydrogen bonds between adjacent chains. Now, each individual hydrogen bond is weak, but when you have many, many hundreds of thousands of millions of hydrogen bonds, it suddenly becomes super, super strong. So it's these many, many hydrogen bond cross-linkages between the adjacent chains of the cellulose molecules that makes cellulose such an important structural material. Let's look at the molecular structure of cellulose. So we can see here, in order to form a glycosidic bond between the two beta-glucose molecules, each adjacent molecule needs to be rotated 180 degrees. So you should notice that the CH2OH branch is pointing in different directions, and you can also see the oxygen pointing in different directions in each one. So we can see that we form that glycosidic bond there. This enables us to form long, straight chains with cross-linkages between them. Cellulose molecules then group together to form microfibrils, which are then arranged into parallel groups called fibers. These groupings provide both strength and rigidity to the plant cell wall. The cellulose cell wall also prevents plant cells from bursting as water enters it by osmosis. It does this by exerting an inward pressure that stops any further influx of water. So when you've watered your plants, the cells become turgid. The cell membrane pushes against the cell wall and exerts an equal and opposite pressure inwards. This causes the whole plant to stand upright. And that's basically everything that you need to know at this point about cellulose. Well done.